Okay, so the acoustic telemetry project on Lake Whitefish that I'll be speaking about this morning is part of a broader two-eyed seeing research study called Together with Gugunyag. Gugunyag is the Anishinaabe-Moan word for fishes. And you can see the artwork that was prepared specifically for our project up here, showing three faces. Those faces represent the three partners on this work, which include Saugeen Ojibwe Nation, Parks Canada, and the Ontario Ministry of Natural Resources. This two-eyed seeing research project includes gathering both Indigenous knowledge as well as using Western science approaches to understand the ecology of Lake Whitefish in this changing Lake Huron ecosystem. There are several parts to this study, one of which includes the acoustic telemetry work that I'm going to share, but we're also looking at other aspects of Lake Whitefish ecology in this project. But today I'll just be sharing about our acoustic telemetry work. So the acoustic telemetry project has several different objectives, the most important of which is to quantify and just better understand the movement of Lake Whitefish around Lake Huron. So between management zones, among the different basins of the lake, and between Canadian and US waters. As you can see here, this is showing the current Lake Whitefish quota management zones for Lake Huron, including those in Ontario waters and in the US. And currently our harvest quotas for Lake Whitefish are allocated amongst these different zones or by the zone. And so I think it's particularly important at this time period to understand movement of Lake Whitefish between these zones because the stocks are at such low levels and there's concerns about sustainability of these populations. So it's really important to understand how these stocks are moving around the lake and being exposed to commercial harvest pressure and other sorts of stressors. So our ultimate goal is to inform management decisions related to commercial harvest and sustainability of these Lake Whitefish populations. As well, a component of this work involves looking at overlap and interactions with lake trout. So we're also doing acoustic telemetry work on lake trout, but I won't be speaking about that this morning. That work is really just getting going now. And so our approach with this project is to tag Lake Whitefish during the spawning season on particular spawning shoals, and then to track their movement throughout the lake as these fish swim around and are detected on various receivers. So this graphics here is showing kind of the evolution of the GLaDOS array in Lake Huron over the course of our study. So our study began in 2020, and it was really this collaborative project that allowed us to rig up, the Ontario waters of Lake Huron with this very expansive acoustic array. So we really wouldn't have been able to do this without that collaborative research. So you can see here just how the receiver array has expanded throughout the course of this project. And so you can kind of keep this in mind as I'm sharing some of the results that, there has been some changes over time in the receivers. Okay, so this shows the Lake Whitefish tagging that we've been doing, focusing on 2020 to 2025. So as I mentioned, we've been going to different places around the lake, and so you can see the numbers of fish that we've been tagging. Most recently this fall, what was quite exciting for us was that we tagged 70 Lake Whitefish down here at Kettle and Stony Point, so down in the southern Main Basin, and we're really interested in learning how those fish are moving around the lake. But that population and some other populations I won't be sharing with you today just because we don't have enough years of data for those fish. So I'll be focusing on some of the populations where we have a little bit more information. Okay, so just to give you sort of a prep for some of the results that I'm going to share. I'm going to be showing you some detection plots by location. And I should mention as well, this, all of the data has been filtered that I'm going to be sharing with you, including, removing false detections and fish that were suspected as being dead or sort of disappeared out of our array, for example. So you can keep that in mind while I'm going through these results. So I'm going to be showing detection plots by location, and this was prepared by Camilla Ryther, a biologist working with us. She also did some residency analysis to quantify, movement and time within these different management zones. I'm not really going to be speaking too much about that, but that work is underway. And then I'm also going to be sharing some results of Sophia Derora's master's thesis. So she's just finishing up her master's at Trent University, working with me, and she's been doing home range analysis to sort of quantify the home ranges of these fish from different populations. And then she was also looking at sort of how much time these fish were spending in different management zones. So I'm going to be sharing some of that work. And so what that home range analysis involves doing is looking at something called, or estimating something called, the center of activity. So that's what this plot down here is showing. So fish can be detected on more than one receiver. And so in a 24-hour period, you get this center of activity for an individual fish. And then you use that to plot home ranges for these individuals. And so specifically, we're looking at 95% kernel utilization density home ranges for these fish. Okay, so to share the results, and I should say this presentation is really kind of like an update on some of our more recent results, and this project is just sort of every year we're getting new information, and so it's very much a work in progress, and we're still learning a lot about these fish. So to begin with, I just wanted to say something about the status of these lake whitefish that we've been tagging and following over time. So currently, so in this data set that I'm going to share with you that goes up to 2025, about 57% of our originally tagged lake whitefish are still swimming around in Lake Huron and being detected on our receivers. We then have 92 fish which were deemed dead just based on the fact that they're just sort of have continuous detections at a single receiver, for example. We had 32 of our fish were harvested. So these were where the tags were picked up by harvesters and reported to us. So those are those fish. And then we have 117 fish, which basically have disappeared from our array, which probably just means that they could have been harvested, but they also could just be in a part of the array where they're not being detected, but they're also not sort of swimming around enough in order to be picked up on these different receivers. So those fish have been removed from our analysis that I'm going to be sharing with you up until the point that they disappeared. So that just gives you a sense of kind of the data that we're dealing with here. Okay, so I'm going to be sharing with you these detection plots for some of the different populations. I'm not going to show all of the populations. And on this side here, and I'm going to be breaking it down by season. So this is the winter, spring, summer, and fall. And so this is looking at a particular population that we're calling Nawash. So this is the tagging location. So you can see the blue star represents the tagging location. And then you can see the detection plot, so where individuals have been detected. And so this is in Georgian Bay, and you can see here that fish are really constrained to very close to the region where they were originally tagged. And so we see no detections outside of Georgian Bay. So on this side, this is just kind of showing a zoomed in version of Georgian Bay where all of our detections are contained. And so you can see, I should mention as well, that the larger the circles, that represents detections from more individuals. And then the color represents the number of detections, with red being higher numbers of detections. So just wanted to highlight a few things. So as I mentioned, so this is the winter. Most fish are constrained within this fairly small region. Same thing in the spring, summer, and fall. So there really isn't a lot of movement even around Georgian Bay for these fish. Okay, so this is showing the home range plots now for that same tagging location. And this is now broken down by year and season. So we have fall 2021, winter 2022, so on and so forth. So it's going in order. And so the colors here represent more overlap among different individuals in their home ranges. Okay, so I mean, this basically paints the similar picture to what we saw with our detection data on the previous plot. As I mentioned, Sophia was also looking at the proportion of the centers of activity of individuals you know, the proportion of their time spent in different management zones, basically. And so what she found was that there's, you know, as kind of the plots speak for themselves, a consistent high percentage of centers of activity are found within the management zone of tagging, so about 77% on average. And then there's a little bit of occupancy in a few adjacent management zones within Georgian Bay, just keeping in mind these are quite small management zones. Okay, so this is Randolph Bay, another tagging location in Georgian Bay. So just, you know, similar plot, so you can see the tagging location as the blue star. And on the right is our kind of zoomed in version. So we didn't see any detections of these fish outside of Georgian Bay. So you can see the detections falling, most of them falling within that area. And then we see, you know, one fish sort of wanders up a little bit more north in Georgian Bay. But again, we see a similar pattern throughout each season. So the winter, spring, summer, and fall. Here's our home range plots for this same tagging location. And here again, we see most of the activity. So fish are really sticking close to the management zone where they were tagged in, with a little bit of use of adjacent management zones. Okay, so now we're going to move into one of our main basin locations called Dorcas Bay. So you can see the tagging spot here up in the central main basin. And so here we're seeing sort of a little more movement than what we saw in Georgian Bay. So we see some fish are moving sort of north. We have some detections further down south. We have detections in the southern main basin as well. But you can see the dots are a little harder to see, and that's because there's not very many individuals moving down into the southern main basin. Most fish are really concentrated around the tagging location. Similar in the spring to the winter. And then in summer, you can see now there's a few more individuals venturing out into basically the middle of Lake Huron, which is quite fascinating, and a few individuals making it over to Saginaw Bay. And then in the fall, we actually see some fish over there in Saginaw Bay as well. This is what the home ranges look like. So basically we see there's more spread among the individual home ranges. And they are using, you know, a few different management zones and intersecting management zones around Tobermory up in this area. And we do see an increased use of management zone of where those fish were tagging in the fall. So they kind of like spread out a little bit, and then we see that still about 76 to 81% of the time the fish are found in the fall within their management zone of tagging. So this is another tagging location in Lake Huron in the main basin. This one's a little further south than the one I just showed you. So you can see the blue star is our tagging location. Again, looking at winter, spring, summer, and fall. So here we see the most concentration is just south of that tagging location, but not very far. We see a few individuals making their way a little bit north, but not quite as much as the Dorcas Bay population. So these fish are kind of tending to stick just a little bit more south than the previous population that I showed you. And then in the winter, we do have some use of the southern Main Basin and over in Saginaw Bay. And then, an individual or a couple of individuals that make their way over into U.S. waters. And then we also see an individual over here in Georgian Bay. And similar patterns for the rest of the year. And here's our home ranges. And, despite the fact that we do have this spread among individual home ranges, again, fish are still mostly residing within their management zone of tagging, keeping in mind that that zone is actually the largest zone. So, maybe that's not too surprising. And then finally, Blind River. So this is up in the North Channel. So you can see the tagging location up there. This is just zooming into the North Channel where all of our detections were found for this group of fish. And so we can see that, there's, an individual that sort of comes a little bit down south. You can see a few more detections down in these areas, but otherwise all of the fish are remaining up in the North Channel. And then here's our home range plot. So 97% of the time fish are within the North Channel. Okay, so just to quickly summarize, so fish tagged in Georgian Bay and the North Channel remained very local to these basins with minimal movement outside of the basin. So almost no fish were making their way outside of these areas. And then we also found that the home ranges were confined to a smaller area, and we saw more overlap among individuals in those locations. In the main basin, a little bit of a different story. So fish did show higher rates of movement, including, into U.S. waters, down into the southern main basin. And the home ranges were more spread out. So there was kind of less overlap among individuals. However, despite this movement that we do see among some of the individuals, we did find that most fish remained within their management zone or tagging, or sort of for most of the time were residing within their management zone of tagging. So as I mentioned, this work is still ongoing, and each year, we're servicing our receivers and getting more information, including on some of the other populations that I didn't share with you. So there's a huge team of people involved in this work. I'm just, kind of the person up here giving the presentation, but people have obviously been working in the field, doing the receiver maintenance. We have tagging crews out, multiple tagging crews out each fall. It's a huge effort by a number of people that wouldn't be possible without our collaboration and wouldn't be possible without Gladys as well. And that's it. Thank you very much.